Today we'll be talking about plant biotechnology. First, we begin by looking at how agriculture has really shifted from a hunting and gathering time during the Paleolithic and Neolithic time to when we hunted and gathered wild food to moving past that time up until industrialization, where food has been genetically modified in some way by making selections, primarily through hybridization and selective breeding. Today, to meet the demands of the growing population, there is a need for increased food supply. One tool in the toolbox of plant biotechnology is to utilize transgenic plant biotechnology. And that involves taking a gene of interest from a non genetically related organism and placing it into a plant for a desired characteristic. And this allows us to move past traditional breeding that relies on genetically related plant organisms to make that cross. And outcomes of this technology have been to develop plants that are herbicide resistant, pesticide resistant, as well as incorporating vaccines. And that's a new technology, and we're not quite there yet. And I'll say more about that in a little bit. Now, with genetic modification, I would argue that we've been modifying genes in the plant genome for hundreds of thousands of years. And this started with selective breeding, choosing plants that had genes of interest. And it may have been through mutation or otherwise, but we've saved those seeds and through that have modified the plant's genome. For example, if you look at that tomato on the screen, the red tomato and that small green tomato, the green tomato is the wild, and the red tomato is what we grow today. And really, it only differs by 3 to 4 genes that were modified to give that red color, the change in size, and sweetness. Now, hybridization is where we make a cross between two parents and create an offspring that has multiple genetic traits of interest. And another characteristic of a hybrid is that in subsequent generations, those seeds from the hybrid do not produce those characteristics again. So they will be able to reproduce, but the offspring will not contain all of the traits. Now, the term genetically engineered or genetically modified organism, or GMO, is the term we use to talk about genetic engineering. And I would say genetic modification has been happening for hundreds of thousands of years, but genetically modified organisms are what's new and novel because we're taking a gene of interest from an unrelated species and inserting it into the plant genome. Now, when we look at hybrids, we can look at some examples here. In a lot of cases, these examples are showing a cross between two dissimilar parents. And it could be a plum and an apricot, or a tangerine and a pomelo. But it could even be something like sweet corn, where we have two varieties of parents that we cross to get the genes of interest all into one variety, and that would be sweet corn. Now, biotechnology in plants has allowed us to advance further than selective breeding or hybridization has up until this point. While the advantages of plant breeding is that it allows for lots of genetic diversity, we can also see that plant progeny, we have vast numbers of plants that produce lots of offspring, but plant biotechnology allows us to overcome the need for sexual compatibility. And plant breeding can take several years, even a decade, to produce a plant with all the genes of interest. And plant biotechnology using genetic engineering allows us to get down to the molecular level. Using that vast number of prodigy and the plant's ability to reproduce from one cell, it allows us to overcome some of the boundaries that selective breeding and hybridization have. And so selective breeding has been the tool of choice for many generations, and we still use selective breeding in a lot of food crops and plant crops alike. And yet we've moved past uh, just selective breeding where we're producing crosses to go down to the molecular cellular level. We didn't talk about protoplast fusion as much in class when it comes to hybridization, so we're going to move past this. But the biggest difference you can see here between plant breeding and plant biotechnology is that the selection of the gene of interest in the parent gets incorporated into the offspring and produces an offspring with that desired trait. What's different in biotechnology is that we don't have to make this cross. We can just take the gene of interest and insert it into the plant genome, and then we can use vegetative propagation to be able to create the offspring. So it could look something like this, where we take just a few base pairs of interest from a gene from an unrelated organism, and we will insert it into the genome. This requires a construct of a promoter, a terminator, and a selective marker gene. Now, how do we insert that gene of interest? We typically use a bacteria called Agrobacterium tumefaciens, and it has this circular DNA where we insert the gene of interest into the plasmid. This bacteria will then be used to wound the plant and infect it. It will create a gall, and in that bacteria, the gene of interest will be inserted into the genome. The plant will then take it up into its own DNA and then replicate it and then produce that trait. There are other ways to accomplish this using Agrobacterium. If we transform that Agrobacterium plasmid with the gene of interest or the genes of interest, we can take leaves of the plant of interest and we can cut out little pieces and place it onto the bacterial plate here with the transformed plasmid with the gene of interest. And then ultimately that plant tissue will take up that DNA, and then through tissue culture, we can produce new plants. We can also use a gene gun. This gene gun will contain tiny tungsten balls or ones that are made of gold that will contain that cloned gene. That gene will be covered all over these particles. You'll load it into the gun and then shoot it at the plant tissue. And then the plants will take up that DNA. I've always wanted to use a gene gun. I have yet to do so. Now, there are more specific ways to target parts of the plant's genome, particularly in the chloroplast. Like mitochondria, chloroplasts have their own DNA separate from the nucleus. And a benefit of using chloroplast engineering is that you can use more than one gene of interest at one time to incorporate into the plant of interest genome. Now, once the plant has been transformed with the gene of interest, there is a whole process of testing and evaluation. Whether we're talking about plant breeding or using genetic engineering, there's a phase of testing that looks at the traits being passed along into the new plant's genome. And so in this case, we're looking at salinity in wheat, and this is the untransformed plant that's not salt tolerant with the transformed plant that is salt tolerant. And this has been going on for many generations of plant breeding. We have to grow up many varieties, or sorry, many plant offspring to ensure that that trait has been adequately fixed into the genome and expressed in the plant. One of the first ways genetic engineers introduced this technology into the field was through the flavor saver gene that was patented and put into tomatoes back in the 1990s. This was done in Harry Klee's lab at the University of Florida. What happened was this polygalactolinase enzyme typically digests pectin in the tomato cell wall and induces ripening. What they did was they were able to isolate that gene and produce a complementary mRNA strand that binds to that mRNA, the normal one, functionally inactivating that mRNA. And so therefore the enzyme wouldn't be produced. And so it delayed ripening and ultimately prevented rotting like it normally would. When the enzyme is present, you would see this degradation. And with this inactivated enzyme, it would delay ripening and extend the shelf life of the tomato by 3 weeks. This created questions and concerns, especially when this technology was new 20 years ago. In this case, it was using the plant's own genome and editing it. We've been able to move further into that area. Using CRISPR-Cas9. And yet, when this technology really opened up, there was thoughts of, oh, well, if we are worried about plants freezing in the field, if it's too cold, we could just take a gene of interest from, say, an Arctic fish that doesn't freeze and put that gene of interest into a plant. And people just started to get worried. What were the limits? What were the boundaries? So really the direction of silencing or editing the plant's own genome through using RNA interference that would essentially silence or inhibit the function at the molecular level became the method of choice and really what's more accepted today. But ultimately, where we started was looking for disease resistance, essentially causing plants to be able to overcome plant viruses or plant diseases, and basically allowing them to take in this antibiotic gene into their genome, which essentially would cure the plant internally, even if it were to be in contact with the virus. Some famous cases of this include the papaya ring spot virus that was decimating Hawaii's papaya population. And back in the 1990s, this work was done by 2 Cornell professors who were from Hawaii. And it saved the Hawaiian papaya industry, which really produces 95% of all of our papaya in the world. Another example is causing plants to produce toxins for pest control. So rather than having to apply more pesticides, the plant is producing its own pesticide and able to fend off plant invaders. So it does this in one way by using Bacillus thuringiensis Bt. Bt is a bacteria that when insects that are soft body consume it, it produces a toxin that kills off those insects. For example, the European corn borer in this stage will be killed off by consuming Bt. Bt is considered an organic pesticide, and so organic users use this, though they're banned from using genetic engineering technology. This BT can then be inserted into the plant's genome. The plant will produce the BT in its plant body, and then when the insects eat it, they will die. Of course, this technology has limitations because there is not an ability for all insects to be killed off by bacillus. And it's non-harmful to people to eat plants that have this BT inside of it. Another option is for weed management for herbicide resistance. So typically glyphosate is used in maintaining crops and reducing weed pressure. So if you look on the left here, this is what weeds look like intermixed with soy. And this is a problem for the soy. It decreases yield as those plants fight for nutrients and space and sunlight. So glyphosate applied to a soybean field that's not genetically engineered will die because soy is subject to the mechanism that glyphosate will kill off the plant ultimately by interrupting photosynthesis. If we insert a glyphosate insensitive gene into the bacteria that goes into the soy, then we can spray the field and it will destroy the weeds. It won't destroy the soy. Concerns with this method were that this could then potentially produce super weeds, right? If you have the weeds akin to the or near the soybean fields or other fields that were transformed, perhaps there could be some of that DNA taken up by those weeds, and then they would be difficult to kill. In reality, that hasn't really come to pass, but there is a risk of resistance. There are some other applications for insects and also increasing fiber strength in cotton, and some others that we'll touch on, including the use of plants for human vaccines. So there have been some trials looking at inserting vaccines into food plants like bananas, potatoes, tomato, to make them shelf stable, to reduce the need for somebody that's trained to give an injection, and also help to administer it in developing countries. One other application of genetic engineering that helps us advance the food crop is through enhanced nutrition. For the longest time, breeding efforts have really been focused on yield and other forms of crop improvement like disease resistance. However, breeding for genetically engineered or genetically enhanced nutrition has really been left behind. And one application of using this technology is to enhance nutrition. One of the famous cases is in golden rice, taking a gene of interest from another organism that produces high levels of beta carotene and inserting it into the rice genome. And why this was done was this improved the nutritional value of rice and even corn to improve it for those that use this as a staple food crop. Because especially in countries where rice is a major staple food crop, there is a high rate of non-accidental blindness due to the fact that many people in that nation are not getting enough vitamin A in their diet. Unfortunately, this work really never took off the ground and is not being used in the food supply, mainly because of social resistance. It changed the color of the rice from white to yellow. It also was difficult for people to accept genetically engineered food. And there are some other issues related to genetic engineering having to do with who produces these patents. Mainly, these are companies that are US owned, or there's only a few in the world that do this work and distribute these seeds, which created a monopoly. And so this was challenging for farmers to not be able to have a food supply that they could save their seeds from. And it also created issues with um, other countries just fearing that the technology, um, fearing the unknown of the technology. In some places, they've even banned the use of genetically modified organisms. And so while there's a lot of research uh, that looks at the health and the safety and the efficacy of genetically modified organisms, there's still hesitation, if not for what the technology produces in the plant, but also the social issues that it has produced. A way to see it in a different light is that uh, these large companies are similar to pharmaceutical companies, where they put an investment into the research and the technology to develop these crops, and some money has to be made from this process, whether we like it or not. So patenting has been a way to help companies regain some of those costs incurred, and uh, and yet there are cases where uh, companies have genetically engineered food crops, especially for issues in developing countries, with the expectation that they would not be paid for this work. So they would do it for free. So that's important to note, because a lot of times it seems that these companies are just in it for the money, but oftentimes they're also using this technology for good. Also, keep in mind that while it's typically seen as industry driven, there are academic institutions in higher education that are also producing these genetically engineered crops. Whoever produces genetically engineered crops still have to go through the same set of regulatory steps here in the United States. And I'll talk more about that in a little bit. Now, if we look to the future, we can see all kinds of ideas on the horizon. In this case, looking at coffee and decaffeinating it prior to it being harvested. You might think, why would you want to do that? On a day like today, I can understand the benefit of caffeine. However, one reason is that the process for decaffeinating beans can be quite hazardous. There's also look at creating food crops or plant crops, I should say, for fuel, for bioethanol, for biodiesel, and other options like in yeast. And one of the things that we've seen since the 1980s is an increase in biofuel production, particularly ethanol. And this continues to be a trend today. We've moved into using ethanol in gasoline as a fuel additive to get away from other more hazardous compounds like MBTE. And yet this means that some of the cropland that was used for food is now being used for fuel production, which in my view is a mixed benefit in that we're not using fossil fuels. However, we're still using biofuels that isn't really getting us away from being dependent on fossil fuels ultimately. And yet the thought of using corn and other plants for biofuels is that it would increase the chance for being carbon neutral. If plants are produced, they consume carbon dioxide, and then even if they're combusted, it would equal the carbon dioxide that's emitted from the combustion would equal the amount that was taken by the plant originally, creating a carbon neutral situation. So there's been research looking at increasing oil production in plants like Jatropha and even in algae. And aside from some of the social issues of, or the economic issues of who benefits from this technology and patenting, There were concerns about human allergenicity and antibiotic resistance. So far, none of those health conditions have come to light. In fact, there's a lot of oversight. There is, however, a very landmark case of Greenpeace discovering that corn that was meant for cattle feed only made its way into the food supply and ultimately into taco chips and corn tortillas for Taco Bell. And so, while ultimately it didn't cause harm, the concern was we need to be careful about the food supply, and what is tested for human consumption should only be for humans, and what's only tested for animal consumption should be kept separately. I think that there's a lot of opportunity to improve the food supply, both for nutrition and also reducing allergenicity in food crops like peanut. Now, the concern originally was impacts to non-target species, the monarch butterfly being one of them. And originally, the research found that there were crops of corn and soy grown close to native plantings of um, milkweed that is consumed by monarchs. And what happened was the pollen from the transgenic crop passed to the milkweed, and butterflies ate that BT pollen and died. So ultimately, they figured out they need to spread out the fields from these native pastures and give a wider berth between these fields so that this pollen cannot make its way to non-target species. Now, as far as regulation, there are different regulatory arms in the government. The USDA oversees uh, regulations about safety as far as growing plants. The FDA focuses on food safety for consumption, and the EPA develops regulations that help evaluate risk to the environment. Plant biotechnology has increased over time, as you might imagine. Some of the major food crops include soy, cotton, and corn. One other thing to note is that one of the more popular technologies of using RNA interference is by silencing genes. And something I talked about in class is the concept of the Arctic apple. Several years ago, it was the apple was evaluated for genetic engineering. And one of the issues in the apple is its browning. And so this RNAi technology was applied to the apple, and it silenced a pathway that normally would produce an enzyme called PPO. And this PPO is what is responsible for the browning. And so the PPO enzyme being silenced meant that the apple was no longer going to brown. And at first I thought this is kind of a first world problem. However, 40% of our apples do not make it to market due to bruising and are wasted before they even make it out into the consumer. And so the thought is that if we can reduce browning, this will also increase the food supply. All right, so that is a walk through genetic engineering and biotechnology.